Good morning, good afternoon, or good evening, no matter where you find yourself in the world. We warmly welcome you to our latest Novogene webinar. In today's webinar, you will delve into the world of transcriptomics. We'll equip you with a clear understanding of RNA sequencing and bring you up to speed with the best practices and the most recent advancements in the field. For those who have been with us for some time, you are likely aware that we've been hosting a series of webinars. We are pleased to remind you that all our previous webinars, including our ever-popular beginner's guides, are free to view on our website. We will send you a link to our website in the follow-up email. A bit of housekeeping before we start. The presentation will run for around 45 minutes, with 10 minutes for questions at the end. We encourage you to submit your questions using the Q&A feature located at the bottom of your screen. You can upvote a question by clicking the thumbs up symbol next to it. If we don't have time to answer your question, we'll make sure to follow up on any unanswered questions via email after the webinar. And please don't use the raise hand functionality if you have any questions or encountered any technical difficulties. Please reach out through the Q&A section or message us directly. We will also share the slide deck and a recording of the webinar via email, which you can expect this on Friday. If you are studying and need a certificate of attendance, there will be a link in the follow-up email that will display a certificate you can print directly from your browser. Finally, there will be a short survey that pops up when the webinar finishes. Please take a couple of minutes to complete this, as your feedback is invaluable to us and help the content of our future webinars. And now I would like to introduce your host for the webinar, our dedicated account manager for Poland, Thomas. Over to you, Thomas.
Yeah, thank you, Miranda, for a kind introduction. So hello, everyone. My name is Tomasz. I'm an account manager at Novogene Europe, and I will be the host of this webinar. Thank you for joining us in such large numbers in this event. Today, my colleague Carolina from Product Management team will discuss about transcriptome sequencing. But before that, let me briefly introduce our company. Novogene was founded in 2011, and since then we are continuously developing our portfolio of NGS and third-generation sequencing, which is why we are present with our services in various segments of science. Like you can see on the slide, we are present in the research, in clinical oncology, and also even in the water testing segment. Sorry, guys. Well, our European Sequencing Center is located in UK, in Cambridge Science Park. The lab is equipped with our own intelligent sequencing platform called Falcon 2, which is increasing turnaround time and excluding human errors during sample and library preparation. We are also up to date regarding sequencing technologies and instruments. So we are already offering to our customers possibility of using Illumina NovaSeq X Plus and PacBio Radio systems. Thanks to our supercomputing capacity, we can process large amount of data and offer to our customers with the bioinformatics analysis. Novogene is a global company. We are not only present in Europe, but also in Asia and the US. It is worth to mention that all our labs are standardized, all are using same Falcon 2 platforms combined with cutting-edge sequencing technologies. We have over 2,600 employees and more than 5,600 customers all over the world. We are providing a full range of next-generation sequencing services, starting from DNA sequencing of human, but also animal and plant samples, We also have our own solid tumor panel of 484 oncogenes and cancer risk panel of 106 genes. We are offering various types of RNA sequencing, which is the topic of this webinar. We are offering metagenomic services and epigenomic services. Almost all of these services can be done on Illumina, PacBio, and then Oxford Nanopore platforms. It is not mentioned on the slide, but please keep in mind that we can also accept pre-made libraries for sequencing. Why Novogene, you may ask? There are many reasons why you should choose us as your sequencing partner. Providing highest quality services is our priority. We are checking your samples on every step. There's a sample quality control, library check, quality check, finally, data quality control. We are increasing our capacity all the time by investing in the newest sequencing technology with the highest throughput. Using Falcon 2, our intelligent NGS platform, positively impacting our capacity and turnaround time. We are flexible. Our labs are accepting your projects on the different stages. Like you can send us samples for the DNA, RNA extraction. You can send us already extracted nucleic acids. We can accept pre-made libraries, as I mentioned. And finally, you can send us sequencing data for the analysis. And finally, product management team and our labs always looking for the new protocols and solutions to provide to our customers. We are very proud that we have been cited in over 16,000 research papers with total impact factor over 10K. It is worth to mention our contribution extends over 69 NGS-related patents that's driving the NGS industry forward. A couple of words about the Novogene Europe. The Novogene community is expanding. We are present in almost every European country. You can get in touch with your local representative to learn how NGS can work for you and your projects. We also regularly host seminars in the universities and various institutions across Europe. So if you would like us to organize a seminar near to you, please do not hesitate to get in touch with us. Briefly, I would like to introduce to you our experienced and very talented European sales team. They are based across the continent and ready to help you with your next sequencing project. So as you can see on this slide, we have very big team in UK and Ireland and the DACH team. We speak your language, so do not hesitate to contact us with any next generation sequencing related requests. Well, that's all from my side. Thank you for listening. Carolina, over to you.
Thank you, Tomasz, for the introduction. And hello, everyone. Just give me a second to share my screen. Okay. Right. So once again, good morning, good afternoon, and good evening to everyone who joined our webinar. Thank you so much for joining. And I'm extremely thrilled to give you an introduction about the RNA sequencing world, unlocking the transcriptome. And I just hope that you'll find this webinar helpful, whether useful, whether you are a researcher, an industry professional, or even a student. Okay, so please buckle up, sit comfortably, and let's start. This is just a general overview of today's talk, and I will start with, of course, introducing what is RNA sequencing and the different methods that we actually have to study the transcriptome. And then we will follow by some tips about data analysis just to understand the usual procedure and the basic steps on how to processing RNA sequencing data. Then I will continue by introducing some latest trends, some emerging technologies in the RNA sequencing world, and we will finish off with a couple of case studies to see the real applications that you could do with RNA sequencing. All right? Okay, so first and foremost, we'll start with the transcriptome by introducing what it is. The transcriptome is the complete set of RNA transcripts that are present in a specific cell or group of cells under a particular set of conditions. So the study of the transcriptome, which is also known as transcriptomics, enables us to understand how genes are turned on and off under a specific set of conditions. So this makes us understand biological processes, development, diseases, and also find new uses in precision medicine, for instance. Now, before we continue, I would like to mention that here in this seminar, we are going to assume that you have a foundation in molecular biology, so there will be some small details that will be omitted, okay? Now, to understand the transcriptome, to study it, first we need to know its key components. So of course, the transcriptome is made by RNA, but RNA can be classified into different types. The most popular one and the most studied one is the messenger RNA, also known as mRNA. And please remember that mRNA in most eukaryotes, they contain a poly-A tail that is well preserved in most cases. Now, aside from the messenger RNA that has the role of encoding the protein, we also have non-coding RNA. And non-coding RNA can actually be divided into two types according to their function. We have the housekeepers that are actively involved in the translational or transcriptional processes, such as ribosomal RNA or transfer RNA. And the main role is the catalytic role that enables the actual process. Now, on the other hand, we have the regulatory non-coding RNAs. And of course, by its name, you can guess that they have a regulatory role. And within them, we can classify them as circular RNA or linear RNA, which is depending on their shapes. And then within linear RNA, according to their size, we can divide them in long non-coding RNA and small non-coding RNA. Now, within the small non-coding RNA, even though the usual standard is to say that it includes all the RNAs that are below 200 nucleotides long, usually the most studied one, it's the microRNA, which has a really tiny size, around 19 to 22 nucleotides, okay? Now that we understand the elements of the transcriptome, how do we explore it, right? There are several methods to study RNA transcripts, and I will start with the most common ones, which is RNA sequencing, our main protagonist today, and it's the high throughput method, the one that allows a full sequencing of the transcriptome and enables us to obtain a very comprehensive view of the transcriptome. So we are able to implement qualitative and quantitative analysis approaches on it. Now, on the other side, we have microarrays, which are which have been with us for a longer time, and they are also able to profile transcripts, but they do it by doing a hybridization of a cDNA or RNA sequence to a chip that contains known sequences of DNA and RNA. And because of this process, which is limited by the probe design, unlike RNA, it's slightly less suitable for detecting novel transcripts because you need to know the oligos when you're deciding it. Okay, so these are the two most common ones. But now there's also another ones. And as you can see here, we have the reverse transcription PCR, which is just a standard process where you are amplifying RNA sequences by converting it to double-stranded cDNA and then detecting it. It is quite an accurate method. However, it's not easy to make it at a large level, so it's not scalable. Now, another method that we have is the nodal blotting method. it's not the first method that you would choose these days, and because it's very, very time intensive, and in comparison to the other ones, a little bit less sensitive. Basically, it requires separation of RNA through gel electrophoresis. Once you do that, you transfer the results to a membrane, and then with the membrane, you do an incubation with labeled probes. So once again, you are relying on known sequences. Now, the last method, it's the quantitative PCR, also known as qPCR, which is quite similar to the reverse transcription PCR, but has an additional step where you are adding labeled probes, which allows you to measure the fluorescence signal during the amplification process. So that's why it allows you to quantify the known cDNA targets. And it's a very great method to validate results. Now, the next slide shows you another way to view what I've just explained in a more simplified format. So here you have a table that compares the different methods, and you can see that RNA-Seq is able to provide you information about the gene expression to detect alternative splicing events. It's a high throughput method, which means that you can work with a large sample size, and it covers the entire genome or transcriptome in this case. It is a very highly sensitive method and allows also to identify novel transcripts. Now, microarrays, on the other hand, they're also very useful for quantification purposes, but when it comes to understanding the structural components, such as the isoforms or maybe detecting novel transcripts, it's a bit lacking on that side. The rest of the methods, which are more traditional, they cannot reach a high throughput level as RNA-Seq or microarray. However, they are really great for targeted analysis when you specifically know the set of genes that you want to work with and you have a small sample size. And it's also a great option when you want to validate these high throughput results. Okay. Now, in the next slides, I'm going to focus on RNA-Seq, which is the core of today's talk. RNA sequencing, or RNA-Seq, is a high-throughput sequencing technique which reveals the presence and the quantity of RNA transcripts in a biological sample at a given moment. So it's basically taking a screenshot of your sample at the time that you want. And once you do that, once you do the RNA sequencing and you have the data, you can choose two different approaches or implement both of them. So on one side, you have the qualitative approach, which is the analysis methods that you're going to use to characterize the transcripts and allows you to detect novel transcripts, alternative splicing events, gene fusions or aberrant transcripts, and also allows you to do some variant discovery on the coding region. So this mainly addresses the question, what are these transcripts? Now, on the other hand, you have the quantitative approach, which is the one that you're going to measure the expression level. So, of course, the most popular thing to study is the gene expression level of your samples. And you can even compare the gene expression level between different sets of samples. So if you have a control group and different treatment groups, you can compare them. And that's what we call as differential expression gene analysis. And if you take a further step forward, once you have the alternative splicing events detected and you know the different isoforms that are present in your sample, you can also do isoform expression level. So this allows transcript and isoform level expression detection. This mainly answers the question, how many transcripts are there in my sample? Now, the biggest advantage of RNA sequencing is that unlike microarrays, it's not limited to the pre-designed probes. So this offers higher sensitivity and of course a much wider dynamic range in detecting rare transcripts or transcripts that are less abundant in your sample, also isoforms, and overall all these tiny subtle changes in gene expression. Now, something that we need to take into account when we talk about RNA sequencing is biological replicates. For instance, in microarrays, it is important to include technical replicates. But when we talk about RNA-Seq, which has a really high reproducibility, it's much more important to include biological replicates, because that's when you can actually use the results to verify if those differential expression genes that you're observing between one sample and the other, between one group and the other, are actually part of your experimental design and they're not caused by external factors. Okay, so there's not a specific number of biological replicates that you need to use. It really depends on a case-by-case level. But we will say that the minimum number of biological replicates you need is 3, especially because you need at least 3 samples to do statistical inferences. Now, in the next slides, I'm going to briefly explain how RNA sequencing works. But before we move on to the technical part, we need to understand that the RNA has many different types, as we've seen before. And it's usually the case that you're focused on one or a few of them, and you don't want the information of the entire total RNA. So here it's a representation, an estimated representation of the total RNA composition in a mammalian cell. And as you can see here, the ribosomal RNA and the transfer RNA, which have a catalytic role and doesn't provide that much information of how the transcription works, they take most part of the total RNA. And this means that eventually, if we want to focus on messenger RNA or other types of non-coding RNA, we need to find ways to reduce this noise or this kind of data that we won't need in our analysis. So how do we do this? Now, depending on the RNA of interest, there are several methods to isolate them. These are the 3 main methods I will explain today. The first one is known as the poly-A enrichment method. So here you're going to target mainly messenger RNA in eukaryotic samples. So because of the feature of these messenger RNAs that have the poly-A tailed, we're going to take advantage of that and just add some oligo-DT primers to capture these poly-A tailed messenger RNAs and get rid of the rest of the RNA. So basically, in a very cheap and fast way, we are retaining the information that we want. With that retained messenger RNA, that's when we can move on with the actual library preparation and get the sequencing data. Now, one of the advantages of this method is that, well, as I mentioned, it's fast, it's cheap, it works really well on eukaryotes, and also it has available as commercial kits ultra-low input solutions. So for instance, even though we usually ask about 200 nanograms of total RNA for RNA-Seq experiments with this approach, there are also special cases where you can even apply the ultra-low input option and work with samples above 1 nanogram. That would be totally fine. Now, the other option is the ribosomal RNA depletion method. When do you want to use this? Well, of course, if you want to target non-coding RNA, all right, the poly-enrichment method won't work, okay? Or on the other hand, you're not working with eukaryotic samples, you're working with either prokaryotic samples or mixed samples from the environment. In those cases, if you want to obtain non-coding RNA or messenger RNA or both, this would be the best choice. So there are different kits to deplete different types of ribosomal RNAs, those that come from bacteria, those that come from animal, or those that come from plant, all right? So you don't need to worry about that kind of specificity. And in addition to that, you also have the option to deplete globin, which is something that you could do for certain species when you're working with blood samples. You also have the option to remove the linear RNA if, for instance, you are specifically interested in studying circular RNA. That is something achievable. And the best thing is that it can also work for FFPE samples. Why is that? Well, if you have FFPE samples and you want to study the messenger RNA, you might think of poly-enrichment because, I mean, I'm working with human FFPE, so they should have the poly-A tail, right? The thing is that the FFPE procedure is quite harsh. So this means that when you're working with extracted FFPE RNA, they're already been quite degraded, so the poly-A tail is not well preserved. So in those cases, it's much better to choose the ribosomal RNA depletion approach so you're not missing any potential messenger RNA. Okay. Now, the third method is slightly different and it doesn't require any pre-treatment like the other ones. This works particularly well for microRNAs and other types of small RNAs, and it's just a simple size selection. So if you're interested in small RNAs, you can do a size selection. At Novogene, we select those RNAs that are less than 50 nucleotides long, and we can just proceed with library preparation after isolating. Okay, so regardless of the method that you choose to isolate your RNA of interest, once you have it, the next step is the library preparation. And it works for most types. So either way, you choose poly selection, ribosomal RNA depletion, or size selection, you are going to prepare the RNA with or without fragmentation into the soluble size for the sequencer. And then you're going to do the cDNA synthesis. You're going to create double-stranded cDNA and then add the adapters to complete the library preparation stage. Okay. Now, remember that regardless if you're doing it with an external service provider or in your own lab, always run a library size and quantification check once the library preparation is over. Okay, why is that so important? Because it determines the sequencing part. So the sequencing strategy, I'm talking about NGS, and in particularly Novogene, we work with Illumina NovaSeq, either the 6000 or X Plus. But the sequencer will need to adjust some settings, and this depends on the library that you're working with and also the library size. So the sequencing strategy, when we talk about Illumina NGS, basically have two types. We have the parent sequencing, which is sequencing from both ends. So you have your double-stranded cDNA, and it's going to sequence 150 base pairs, for example, on one side and 150 base pairs on the other side, which means that in total you can cover 300 base pairs. Now, on the other hand, you also have the option to just sequence from one side, which is known as single-end sequencing. And you can just sequence maybe 50 base pairs on one side, and that will be the information that you get. Okay, so general practices is that for messenger RNA-Seq, non-coding RNA sequencing libraries, just basically any RNA sequencing solutions that exceed 200 nucleotides, we recommend to do parent 150. If, for instance, you prefer working with parent 100, that's also totally fine, all right? Now, with small RNA libraries, when you tend to focus on studying the microRNA, it doesn't need to be that way. You can just apply single N50 to cover those insert sizes that are below 50 base pairs, all right? And now I just want to mention that when we talk about insert size, it's not the entire fragment size that includes your double-stranded cDNA and the adapters. We're only talking about the cDNA. All right, so please pay attention also to these differences in the terms. Okay, now we are done with the explanation of RNA sequencing, and we also showcase some other techniques. And I finished with the sequencing part. So now we are going to assume that we have raw RNA-Seq data. What are we going to do? We need to process it. Okay, so here you have the general workflow, and we've gone through library prep, we've gone through sequencing. Let's see what should we do with the raw data, which is the data QC. Now, you have different ways to understand how the sequencing results were, and one of them is data filtering. This is the process where you're going to use a specific software and check how many of those sequence reads are actually high quality reads, which are what we know as clean reads, and how many of them contain a high percentage of non-identified nucleotides, which are the ones in yellow containing N. And also you can see low quality, which means that are below Q20, or potential reads that contain adapter sequences. Unfortunately, the example that I showed came from a really nice high quality sample. So you can barely see the colors, but at least the legend showcase the different scenarios. Now, aside from data filtering, you can also do an error rate check, okay? Even though this graph seems like showing a huge error rate, if you check the numbers, it's actually very small. It's talking about error rates of 0.02% and 0.04%. Okay, and in the x-axis, you can see the position along the reads. So this is an example of a parent 150 sequencing project. And because of that, we are sequencing 150 on one side and 150 on the other side. And we are using reagents during this process. You can see that the error rate has a slightly increase once we are reaching the end of the read, but that's totally fine. Now, aside from that, you can also check the AT-GC content, which should be at least similar between them, the AT and the GC. Just a general guideline, at Novogene, we use the FastP software for data QC, and it's very simple. You just need to input the FASTQ files in raw format, and it will just deliver the clean reads, also in FASTQ format. Okay. Now, we're done with data QC, so the next slides will be about the bioinformatic analysis part. We won't go into much details, but it's just to showcase you the different steps that we could. Now, you have the clean reads in FASTQ format. The next thing that you want to do is mapping. Mapping to the reference genome of your species. This means that the reference genome file needs to be ready and in FASTA format, okay? Now, the software that we are advising here is HiSat2 because this is the software that we use in our pipeline. However, there is no the perfect or the only one alignment software. It really depends on the project. It really depends on your computational resources because some softwares, they work super fast. They work really well when you have limited resources. On the other hand, you have those that are aiming at accuracy. Okay, so please don't just choose HiSat2 because it's shown in this slide. You can always choose another one that fits better your own project. And once you input the FASTQ files with the reference genome, the output will be some BAM files, which is the binary version of a SAM file. These BAM files, you can visualize them later with different tools, such as the Integrative Genomics Viewer. Okay, and on the right, you have a picture on the IGV software. Now, once you have the BAM files, the next step with RNA sequencing data is the quantification step, right? You want to count how many transcripts are there. And you start by generating the gene counts. The software here, the one that we use is feature counts, but you have other software available in the literature, and that's completely fine. You just want to make sure that the software that you use can actually accept the BAM files that you generated. And in some cases, they will need a GTF file, which is the annotation file that can provide you information about the gene ID, which is what you can see on the right picture again. Now, once you run the software, you will obtain an output file, which is a count matrix, which is usually in a table format with genes as rows and samples as columns. Okay, you have the gene counts. Does this mean that you can immediately do the quantification or the gene expression comparison? No. Why? Because you must normalize the gene counts first. When you're doing NGS approaches, you're going to generate a different sequencing depth in each sample. So you must do a normalization step before doing the differential gene expression part. Here we use DSeq2. You have other softwares available, and basically you input the raw counts. And in the case of DSeq2, it integrates the normalization step inside, and then it will deliver the results with either FPKM or RPKM and the DGA results file. If instead of FPKM and RPKM, you prefer to work with TPM, that's totally fine. Just know that the DSeq2 software does not provide them. Now, you have the DGA results. What's next? That's the time when you actually get to play and make figures. So you have your control group, your treatment groups, you find the differential gene expressed genes that are there, and maybe you can represent it through a volcano plot. So you can see when you compare the groups, which ones of them are upregulated genes as the ones shown in red, and which ones are downregulated genes as is shown in green. Now, another thing that you could do among all the other choices that you may have is to represent them in Venn diagrams. So you want to see the co-regulated genes, the genes that are present in both group 1 and group 2. So this is just an example for an experimental setup. You have group 1 with one treatment, group 2 with another treatment, and a control group. So you're going to find out the differential expressed genes of group 1 when you compare it against control, and the differential expressed genes of group 2 when you compare it against the control group. Now, once you have those sets, that's when you can make the Venn diagram and compare them, okay? Now, what else? It's cool to compare the DGEs through those figures, but you need to also understand what those DGE means, right? Like, why do I see a differential gene expression between my treatment and my control group? You need to assign a biological value to those genes. And that is done through a process known as functional analysis. And there are many other synonyms available. Functional analysis, enrichment analysis, gene functional annotation step, they are most likely the same. Okay, so you're going to identify those differential expressed genes, in which pathways they're involved in, and what kind of networks they're actually forming. So here, the software that we're using is known as Cluster Profiler. And once they do the clustering, they can actually compare it through different databases. And at Novogene, we use by default the gene ontology and also the key GG, because they are the most well-known, they have a long-standing curation, and they also offer information about a wide range of species. However, if you're interested in something else, let's say the disease ontology or the reactome pathway, that's also something that you can include or ask us to include. That's totally fine. Okay, now, aside from the quantitative analysis that I have been explaining for now, you can also do the other approach, right? You can do some variant discovery, which is basically using the map files to do some variant calling and annotation. And you will have a table like the one that I'm showing here, showing you the different SNPs and different indels. And on the other hand, you can also check for alternative splicing events. So here we're using the software RMATs, and it's designed to detect differential alternative splicing events in RNA sequencing data that has biological replicates. So you will be able to find the expression. So as you can see here, the one in red shows the isoform when there is a skipped exon, and the one in orange shows you the expression level when the exon has been included and not skipped. Okay, so this is just a general overview of the data analysis. And remember, you always need to start with data quality control, no matter what, always do that. And then you need to do the mapping to get the BAM files. And then with the BAM files, that's when you can spread across and do all sorts of downstream analysis, such as the quantitative analysis and the qualitative analysis. Okay, now something important to mention is that what I showcase right now is quite a generic pipeline, okay? And it could potentially work for both messenger RNA and non-coding RNA. However, because messenger RNA and non-coding RNA, they have different biological functions, it is better to have separate pipelines for them, okay? So for instance, in the quality control and normalization steps, you can perfectly share the same software and the same steps, but then the downstream analysis, they must be different. Why? Because messenger RNA, they tend to focus on understanding how many transcripts are there, to see the differential gene expression, and to do the annotation of those differential expressed genes. So if you're interested in that, because I haven't explained that much in detail, you can click on the link that I'm showing below. You will get the slides after the webinar and get access of another webinar that explains this. Now, on the other hand, if you're focused on non-coding RNA, quantification is not a first priority. You want to identify what kind of non-coding RNAs are present in your sample, and then perhaps do some even special pre-processing, like the case of microRNAs. And after that, you might want to study the secondary structure of the microRNAs. You might also want to study the target prediction level, and then perform some functional analysis. So again, if you're interested to know more, we will provide you a link to access non-coding RNA sequencing results and how to understand them. Okay, so we've seen how RNA sequencing works and also the data analysis pipeline, but it not only stays like that. Like, you don't need to work with RNA sequencing data alone. You are able to integrate that data, this transcriptomics data, with other omics, such as genomics, proteomics, metabolomics, and so far more. The main goal here is to understand and obtain a holistic view of the biological system, all right? So, for instance, if you combine transcriptomics with genomics data, it will allow you to explore the impact of the different genomic mutations, such as somatic mutations, such as copy number variations, on the gene expression pattern of your samples. Now, another thing that you could do if you're studying epigenomics and transcriptomics together is to correlate those DNA methylation patterns, or maybe those chromatin accessibility patterns with gene expression levels. That makes you understand how the regulation process can affect directly on the expression of your samples. Now, what else? We can do transcriptomics with proteomics together. This helps you to assess the impact of different isoforms, on the different alternative splicing events on the protein isoform expression. And then my last example here would be combining and integrating transcriptomics information with metabolomics, where you're able to correlate gene expression changes with alterations in metabolite levels. Okay, so we are done with the most technical part, I would say. And now let's just dive into some emerging technologies. I'm going to give you three different emerging technologies, and we'll start with single-cell transcriptomics. So you've probably heard of it because it's been quite a hot topic these days. And single-cell transcriptomics enables gene expression profiling of individual cells within a heterogeneous population. This is one of the main differences between the single cell and the standard RNA sequencing, which is also known as bulk RNA sequencing. So here, this is a figure obtained from 10x Genomics website. You can see that we have a tissue, and usually when you're doing the standard RNA-seq approach, you're just going to extract the total RNA of that tissue, which is the bulk RNA input on the bottom, and then perform an average gene expression analysis. So this means that you're not able to tell parts which particular cell type is being expressed more than the other, or which expression pattern they have, because they could have unique expression patterns. Now, with single-cell analysis, they're able to capture the transcripts that are present within each cell. So when you do the analysis process, you can actually reveal the expression profile of each cell type. This means that you can tell the different expression heterogeneity among thousands of cells. Okay, so this is quite an innovative solution, but my main goal here is to explain that even though it's a really cool solution, it doesn't mean that you must use it, because it all depends on your project goal. Okay, so here's a table showcasing the differences between single-cell RNA-seq and bulk RNA-seq. So on one hand, you have single-cell RNA sequencing that is able to reach gene expression detection of individual cells, while the bulk RNA is just the whole chunk of cells from that tissue. Now, single-cell RNA sequencing is very sensitive to capture expression profiles of those cell types that have quite a low expression level, while, of course, the standard RNA-seq is quite limited. Not only that, with single-cell RNA sequencing, you can even see the changes in gene expression over time, and there are specific pipelines already developed for that. But now, with that, it comes a cost, which is the computational challenges. It's quite intensive to process this large number of data sets because you have RNA-seq data sets of each individual cells, of thousands of cells that are within your sample. But however, it can be extremely useful if your goal is to study cellular heterogeneity, especially interesting when you're studying tumors. And it's just overall one of the first NGS things to think about when you're in the immunology field and the neuroscience field. All right. Now, the slightly downside is that it's more expensive than bulk RNA-seq, of course, because it's an emerging technology and it's also quite tricky to perform it, and it's time-consuming. But it works extremely well when you have a small population of cells where it is essential to identify the gene expression level at individual level. Okay. Now, does this mean that bulk RNA-seq is outdated? Not at all, of course. The application of bulk RNA-seq, it's mainly to study general trends. So you want to obtain an overall gene expression profile of that sample. Okay, so if you really don't care about the cellular heterogeneity, bulk RNA-seq works totally fine. Okay, now, after single-cell transcriptomics, we have another breaking technology, which is spatial transcriptomics. This combines the traditional transcriptomics with the fact that now, by integrating some other features, we can understand and know the position of the cells within the tissue. Okay, so this is quite an innovative technology. And you can see here just a figure to represent the different methods that we have. And how does this work? So first, if you choose an NGS-based approach, you will need to first make a capture of the tissue, and then you will be able to barcode the different transcripts according to the location. Okay, you will insert certain barcodes that will be able to tell you the exact location of that cell of that sample. Now, another approach that you have is called imaging-based, which is also known as in situ approaches. So here you don't need to do any capture. You're able to detect target sequences by, you're able to obtain the transcript sequences within the tissue directly on it. But no matter what approach you choose, in the end, you will end up with a matrix. So this gene expression matrix is basically information about the genes and its corresponding locations. So again, sounds super exciting and can be extremely efficient if it meets your research goal. So again, we have this table and it's going to show you the differences. Spatial transcriptomics works extremely well when you're aiming at understanding the spatial resolution. It preserves the tissue architecture and it helps you understand the interactions between different cells. Now, the challenge here is that it's quite an innovative method and it requires specialized bioinformatic tools to integrate all that information, but it's very handy when you're interested in spatial heterogeneity, when you want to study tumor microenvironments and also developmental processes. Not only that, if you're, for instance, working with very complex tissues, such as the brain, it also helps you to bring a lot of insight to understand the spatial position of your samples. Now, when we compare the cost and the throughput with the bulk RNA-seq approach, it is more resource intensive, which means that it's also slightly more expensive and time consuming. But it answers the following questions, which I've already mentioned: cellular interaction, tissue organization, and spatial heterogeneity. Okay, so our last emerging trend here, it's known as long-read sequencing. Okay, some of you might already be familiar, but they are a more recent technology than NGS, and they keep thriving to bring new applications day by day. So long reads, in comparison to the NGS RNA sequencing, they're able to cover the entire transcripts and enable full-length transcripts. So they address some challenges that short reads have. So, for instance, as you can see in this picture, with short reads, we are disrupting the natural connectivity of those transcripts. We are accommodating the RNA to our needs to be able to sequence it. With long reads, you don't need to do that. So we are reducing the fragmentation of the transcripts, which means that we can improve the isoform detection. And with that, this is just like a butterfly effect, we can detect more accurately different alternative splicing events. So this comes very handy when you want to improve the characterization of the transcriptome. And it helps you to identify better novel and fusion transcripts. Now, just an example of long read platforms, we have PacBio and ONT, which are the most known ones. And PacBio recently has introduced the Revio system with some exciting announcements on RNA kits, and Oxford Nanopore with the PromethION. They are also constantly trying to improve the accuracy of the base calling results. Okay, so hopefully this is the last table that I'm showing you today. This is just a comparison between long-read RNA-Seq and short-read RNA-Seq. So main difference, of course, is on the read length, which plays a crucial factor in allowing full-length identification of isoforms with this technology. Now, the downside here is that in comparison with NGS technologies, the error rates can be higher, not always, there's some exceptions these days. And there are also challenges when trying to assemble these different transcripts because it requires specialized software. While on the other hand, with the Illumina technologies, which have been working on the field for, I'd say, at least a decade, we have very well-established software where we don't need to use a lot of computational resources to proceed with the analysis. And mainly, just for you to have a take-home message, long-read RNA sequencing is extremely effective when you want to improve the genome annotation and also enhance isoform discovery, while short-read RNA sequencing is still very effective to do gene expression quantification, differential gene expression analysis, and also, of course, the functional analysis part. All right? Now, luckily, with long-read and short-read sequencing, you don't need to choose one or the other. It's actually quite common to see a combined approach. And when you do that, you're able to bring out the strengths of each technology and complement each other. So with that, I would like to move on with the case studies and applications. And of course, now taking advantage that you still remember the long reads part, we'll start with building a reference transcriptome with PacBio Iso-Seq and Illumina sequencing. So this is a publication published in 2021, where Novogene had the honor to offer their services. And basically, they were working with a species which had a quite hardly identified genome. So it was hard to get information about the transcriptome. And they decided to try the PacBio sequencing. So they use PacBio isoform sequencing to develop a high-quality reference transcriptome, and then also produced some sequencing reads with Illumina to combine them together and improve the overall quality and reduce the error rate. All right. So first and foremost, they extracted high-quality RNA to perform the experiment. They do sequencing with short reads and long reads technology. And then in data analysis, they did the PacBio assembly, Illumina de novo assembly, and combined assembly. So basically, they compared the different results. And as you can see here, the combined transcriptome was the one that produced the higher number of transcripts that are matched with the BOOSCO results, and also the one with less missing transcripts information, while the de novo only transcriptome, which is Illumina only, and the PacBio only transcriptome showed slightly worse results. Now, on the other hand, when they move on after the assembly to the functional annotation stage, they were able to see that the combined approach also gave more information about non-coding RNAs, so they were able to detect a higher number of non-coding RNA transcripts. Okay. Now, another case study here. It's the multi-omics integration. So here, what we are showcasing is a study where they were trying to identify the epigenome landscape of renal cell carcinoma while knowing that both genomic and transcriptomic landscapes were well understood. Okay, so basically they worked with 2 different patient cohorts and 2 different experiments, including RNA sequencing, ChIP-seq, and ATAC-seq. And within the analysis, they performed separate analysis for RNA-seq and ChIP-seq, and then they integrated the results. And the results showcase that it was possible that with this integrative approach to identify a set of transcription factors that will be involved in regulating the genes that establish the cell identity and function in renal cell carcinoma. So you can see here the different results. This is an overview of the approach here in figure A. With combining ChIP-seq and ATAC-seq approach RNA-seq data, they were able to obtain this heat map through the meta-analysis results. And basically, they were able to identify transcriptor factors that were specific to a certain histology of the cancer cell type. And then to further validate these results, they also did an additional experiment to overexpress certain transcription factors to see that indeed, by overexpressing this transcription factor, the histology, the transcriptional phenotype could change from one histology to the other. So basically, the conclusion was that by integrating the multi-omics data, they were able to find much more useful information than by just targeting the ChIP-seq data alone. Now, just to finish, I would like to introduce you about some Novogene RNA-seq solutions, because we've seen a lot about RNA-seq. And here, just to showcase that if you're interested in studying gene expression, you can always choose messenger RNA-seq, or you can always also tackle the non-coding RNA-seqs on one hand. And if you're interested in obtaining a quality reference transcriptome, then you can either choose IsoSeq or the combined approach of Illumina and IsoSeq. We also have other services, but I'll leave it for another day. So yeah, thank you. And now we are going to give 5 to 10 minutes for a Q&A session.
Thank you, Carolina, for your informative and very detailed presentation. Now we will answer some of your questions. If we won't answer your question this time, don't worry, dedicated account manager will get back to you soon. So yeah, let's start with the first one from Prinas Mahdavi. Determining the alternative splicing via RNA-seq means that we will be provided with the all alternative splicing of the all genes in our sample?
Yes. So when you're performing the alternative splicing analysis, you're working on the BAM files, and then with the corresponding software that you choose, it will show you all the different splicing events and the isoforms that they have found that meet those events, and also the expression level. So you will not only find the transcript information, but also the expression information of all the RNAs that are included in your data. Yeah, thank you.
Perfect, thanks. Another one is from Ramin Sadri. Some non-coding RNA have poly-A, so the mRNA capture by oligo-DT does not just capture mRNA. Is there a method to exclusively capture mRNA?
That's a tricky question. Yes, there are non-coding RNAs that have poly-A tails, but it's also questionable whether a non-coding RNA it's really a non-coding RNA, because there's some non-coding RNAs that are found later to have coding potential. So we are sort of in a gray area. I won't say that there is a specific technique to isolate only messenger RNAs, but then maybe during the annotation step, you will be able to exclude and filter out those that you're sure that don't have a coding function. So in terms of library preparation, I don't think there is a specific solution, but then when you're processing the data in the bioinformatics stage, you could be able to separate them better.
Okay, thank you. Yeah, there's also a question from Songbin Han. So with variant calling, it would be possible to get all SNPs of the genome-wide coding regions, which will be equivalent to exome sequencing?
Yeah, well, unfortunately, it's not the genome-wide, but the transcriptome-wide. So we are using RNA-Seq data. And of course, if, for instance, we are doing messenger RNA sequencing, that information comes from the coding part of the genome. So we are not covering the non-coding areas. It's only the coding areas. So it's a partial view of the variants. If you're interested in seeing the whole genome, it is still better to choose the whole genome approach and work with DNA instead.
Yeah, thank you. And another one is from Davide Clementoni. Could you give me some advice regarding coverage Giga produced for mRNA and non-coding RNA sequencing?
Yes, thank you. This was also mentioned in other webinars, but of course, I'm glad to mention it again. If you're working with messenger RNA sequencing, usually between 6 gigabases to 12 gigabases, it's enough to do the overall view. But of course, if you're particularly interested in a set of genes that you know in advance that have low expression, you can always aim at a higher sequencing depth. And now, when you're working with the non-coding RNAs, the thing is that because I explained in the methods, the ribosomal RNA depletion is not only going to give you non-coding RNAs. You usually have non-coding RNA and messenger RNA. So in that case, you need to double the sequencing depth. So instead of 6, I will say to start with 12 gigabase and then aim higher as you think it's needed.
Yeah, great. And I think the last one is from Monika Migdałek. I wanted to ask about the normalization step. How exactly is it done? How do you choose a method, and what factors do you take into the consideration?
Okay, yes. So the normalization step, when we are working with parent sequencing data, we usually choose the FPKM, which is the fragment per FPKM fragment per kilobase length per million read. All right. When you're working with single end data, then that's when you can either choose the RPKM or also the TPM. And the issue here is that TPM, it's better in terms of that it normalizes the gene counts of the whole sample. So you're able to compare it within samples. And with FPKM and RPKM, you're not able to do that. However, then afterwards, you not only need to use, like the normalized information is not used directly to compare samples. The software that you choose will apply another algorithm to perform the differential express analysis. So it should solve that problem. And I'm not really sure if I answered all the questions, but in any case, I will review it later once the webinar is over, and I'll get back to you on a written text.
Yeah, I think you just did. So thank you. I think we don't have enough time to answer more questions.
Okay, sorry. Well, just allow me to give you the final slide. Once again, I want to thank everyone for attending this webinar. I really hope it brought you some new information. If there's anything else that you want to know, feel free to check out our website by scanning this QR code. It will take you to our webinar page, and you can check the different topics we have. If you have any suggestions about the next webinar topics, or if you have any questions about the services that we have, feel free to contact us in that email address. Or if you're not interested in that, just want to know if we have any sort of fun activities, you can also follow us on social media. And last but not least, I just want to mention that now that we are reaching the end of the year, we have some offers. So if you're interested, please feel free to get in touch with us, and our account manager will reach out to you. Thank you.
Yeah, thank you all for your time today. We hope you enjoyed the webinar and found it useful. Thanks for all the questions. As I mentioned, if we haven't managed to answer your questions live, We'll get back to you via email next week with the answer. We will also email you with a link to the webinar record and the slide deck. You can expect to receive this on Friday. When the webinar finishes, a survey will pop up. We would appreciate you taking the time to complete this, as it helps ensure we make future webinars even more relevant to your research goals. There will be a link in the follow-up email for to view the certificate of attendance if you need one. This can be printed from your browser window. In the meantime, you can follow up us on Twitter, LinkedIn to keep up to date with the future webinars, offers, and news. We look forward to seeing you all on the next webinar. Thank you, guys.
Thank you.